So today we'll be discussing DNA replication. I'm sharing my screen. Are you able to see the questions?
Yes.
Okay. So we'll start by tackling question one, which says outline the central dogma of molecular biology. Uh, is there anyone that has an idea as to what the central dogma of molecular biology is? We'll go ahead. When we talk about the central dogma of molecular biology, this is what we mean. From our previous discussion, we were talking of nucleic acids, and we mentioned to say that there are two types of nucleic acids. There's DNA and there's RNA. DNA, from the previous discussion, we deduced what the structure is according to Watson and Crick. And then we went on to say uh, DNA is capable of forming new strands by replicating itself. And RNA is made from the DNA, whereas the proteins are made from the RNA. So DNA to RNA, RNA will be synthesized from DNA by a process known as transcription, whereas proteins will be synthesized from RNA by a process known as translation. And then DNA will replicate itself. And this outline here, this which I'm showing in this blue box here, is what is known as the central dogma of molecular biology. The next question says, explain the experiments which provided evidence in support of the mechanism of DNA replication. On the experiments which provided evidence in support of the mechanism of DNA replication, if you recall, uh, if you're able to see this image, Watson and Crick, using experimental data from Hawkins and Franklin, they they came up with a model. They deduced the model for DNA. So when they came up with this structure, they thought to say this structure must come together in a way. So there must be some sort of separation and then complementary base pairing to put it all together. They didn't really know how these bases separate together, whether it's in that way, whether they break apart and then form. So uh, after the discovery of the model, the structure of DNA, the scientific community came up with three models on how this DNA replicates. So you have this double-stranded DNA. One of the models which they came up with was known as the conservative model. And in the conservative model, you have this DNA here. You have this DNA here. So in the conservative model, the idea was that at the end of the replication, you have two DNA molecules. You have one which will contain the original strands, and then you have the other DNA molecule which will have two completely newly synthesized strands. The other model which was suggested was the semi-conservative model. In the semi-conservative model, uh, after the replication, you have two strands, two DNA molecules. And in those two DNA molecules, you would have one strand from the original DNA molecule, and then you would have a newly synthesized strand. And then on the other strand, on the other DNA molecule, you would have the same one odd strand and the newly synthesized strand. The third model that was suggested was known as the dispersive model. And in this model, what was
Before you go further, I have a question
yes, go ahead.
about the semi-conservative type
Yes,
of replication.
yes,
When we say the odd strand and the new strand, is it the odd parent strand
okay,
and the new daughter strand, or how do they put it?
okay. So this is what I mean. You have this DNA molecule. So you have this, you have this strand here, and then this strand in the semi-conservative, it separates. So this is the parent strand. This is the parent strand.
Okay.
When it separates, you have one half of the parent strand, and then you would have a new daughter strand. Just a minute. Okay. So you'd have a new daughter strand. That you'd have this here, conservative. You'd have this synthesized, which is the daughter strand. You have this half coming from the parent strand, and then you have a newly synthesized strand, which is the daughter strand. And that was what the semi-conservative was. You have the parent strand split in, and then each half of the parent strand will be bound to a new daughter strand. So you get, if you can see here, you have this gray, these gray strands. These are the parent strands. And then after replicating, you have the two DNA molecules. You have two DNA molecules. Those two DNA molecules will contain an original strand from the parent DNA molecule, and you have a newly synthesized strand. So the third model which was suggested was the dispersive model. And in the dispersive model, uh, it was more like when the new DNA molecules form, the mixture of the old the parent strand and the newly synthesized strand. So you'd have a part containing the parent strand, a part containing something that's newly synthesized. It was more of a patchwork where you have the mixing up. You have the original strand, the new strand, the original, and so on, as represented in this diagram over here. So,
Kindly just explain again on the dispersive.
so in the dispersive, you have this strand. This is the original DNA molecule. So after replicating, uh, the dispersive suggested that after replication, you would have a hybrid strand instead of, if you compare the conservative and the semi-conservative, where you have in the conservative uh, you have one DNA molecule containing just the original strand, and then you have another DNA molecule containing newly synthesized strands. But in the dispersive, what was suggested is that instead of having this kind of arrangement as seen in the conservative and semi-conservative, you would have both strands being hybrid, whereby the DNA molecule who contain bases, nucleotides from the contain nucleotides from the original DNA molecule, and then there'll be newly synthesized nucleotides as well, like that, like so. Unlike having a situation where you have a completely different strand being synthesized, In the dispersive, you'd have say maybe, so in the dispersive, you'd have, let's say you have this base, I have a nucleotide from the original strand, and then the next nucleotide would be newly synthesized, and then you'd have again another one. You'd have them alternating in some sort. So you'd have a new a nucleotide from the parent DNA molecule. Then you'd have a newly synthesized added nucleotide, and so on. So it was like the DNA molecule, the two DNA molecules were being patched together, mixing the original strand and the newly synthesized strand. Is it making sense?
Yes.
Okay. So after these models were suggested, uh, Meselson and Stow, they carried out an experiment to try and determine which model uh, DNA followed during replication. And what they did is they used E. coli, which is a bacteria. And since E. coli is able to replicate faster, so it was more of ideal for the experiments. So they used E. coli and then they grew it in uh, in nitrogen. So you know nitrogen as isotopes. So you have N15 and then you have N14. N15 being the heavier one. So they grew the E. coli in N15 isotope nitrogen. And what happened is that if you remember from the structure, in the structure of DNA, you have nitrogens and so on. So during the replication, the DNA of the E. coli incorporated uh, the heavy nitrogen. And after the replication, after new cells were formed, that was the first generation. Uh, so you had E. coli, which was grown in N15 media. Then this E. coli cell produced a new cell, and then this again was then grown in this was then grown in N14 media. So after the first E. coli cells were grown in the N15 media, the new cells that were produced, Meselson and Stow they extracted the DNA and then they put it in a centrifuge using cesium chloride for So they put the extracted DNA in a centrifuge, and what they found is that after centrifugation, the first cells that were produced, they had a hybrid DNA. So instead of having it, if you look at the bottom, when they centrifuged, they found that after centrifugation, like I mentioned, you have N15 and N14 isotopes of nitrogen. So when you centrifuge them, they will settle at different levels of the test tube. So you have the lighter one, the N14 settling upwards, and the N15 settling upwards. So when the cells of E. coli were grown in this N15 media, and then they did the DNA extraction and analyzed it and put it in the centrifuge, what they found is that instead of being, say, at the bottom where at the bottom where N15 is supposed to be, or at the top where N14 is supposed to be, it was somewhere in between the two levels. This suggested that a hybrid DNA was formed. So meaning a strand was formed which contained the N15, and then the odd one which contained the N14 was already there. So you have this strand here, this one made of N14, then you grow it in N15 media, and what you have is that the new strand, the red one here, the new strand here, the red one shown as red there, would have the N15 nitrogens, whereas the odd strand would have the M14 nitrogens. And this gave it that uh, density after centrifugation. Then when they did the replication now, second time for the stem cells which were produced, they found that they now had two bands where they had one which was at the top and one which was in hybrid position. So you have this one here now, which contains both N15 and N14. And then when this one replicates, what they found is that, so you have this and this blue one separating, and what formed was the one which was containing N15, and then another one. So they, as I was saying, Mesoson and Stow, they grew the E. coli in N15 media, and after the first generation, and they did the centrifugation, they found that the density of the DNA they had extracted was representative of the semi-conservative model. Uh, the, the density suggested to say there was one strand which formed with the odd N14, and then there was a new strand which formed with the N15 nitrogens. And then after the second generation, there are two bands, one containing the hybrid, the one they observed in the first generation, and then the other one which contained just the light N14. After after what they observed in the second generation of cells, they determined to say DNA replication follows semi-conservative model. And
So I have a question.
yes, go ahead.
Sir,
Go ahead.
I didn't get you clear on. I'm saying I didn't get you clear on the reason of why they put DNA N14. What the reason why?
Ah,
My question is like DNA
yes.
were put in N14, 15.
Mm-hmm.
Why? I didn't get you clear that. What is the reason?
What is the reason? So, if you remember the structure of your nucleotides, so in your structure of your nucleotides, you have a nitrogenous base there. You have a nitrogenous base there. So E. coli, say this is a petri dish, and then in that petri dish you have a solution which contains N15. You have a solution which contains N15. So the equali will utilize the N15 in the solution when making new cells. And what you have is that the new cells in that nitrogenous base, instead of having the N14, what will be incorporated will be the N15. And so when they analyzed the DNA of the new cells that were formed using centrifugation, they found that, so you have this test tube here, the isotopes N15, you'd have N15, if you just get them just the way they are, you'd have N15 maybe somewhere there, and then you'd have N15, which is heavier than N14, somewhere at the bottom there. So when they extracted DNA from the new cells, and then they did the centrifugation, instead of having it at the level of N14 or at the level of N15, they had it somewhere around there, which seemed to suggest that the newly formed DNA was a hybrid, which contained both N14 and N15. Okay, so that was about the Mesoson and Stow experiments, which I was trying to explain following question 2, which says explain the experiments which provided evidence in support of the mechanism of DNA replication. Okay, so we'll move on to the third one. Differentiate between conservative and semi-conservative mechanisms of DNA replication. I think when explaining the experiments, I also explained on the different models which the scientific community came up with after the structure of DNA was discovered, which were the conservative, the same. So we've tackled question 1, 2, and 3. We talked about the models of DNA replication that were suggested, which were the conservative, the semi-conservative, and the dispersive. And amongst those models, the experiments proved that the semi-conservative was the mechanism which DNA followed in replication. We go now to question 4, which says, describe the various stages of prokaryotic DNA replication. All right. So in prokaryotes, which are organisms like the one which were used in the experiments, E. coli, and so on, replication is simpler than in eukaryotes. So for the purpose of this discussion, I've sort of divided the replication process into 6 steps, and I'll try and discuss them. So the 6 steps can be thought of as being initiation, then there's DNA denaturation, priming, then there's elongation and proofreading. Then you have separation of the synthesized strands, and then you have termination of the whole replication process. So in the initiation of replication, in E. coli, I'll use E. coli as an example for the prokaryotes. So in E. coli, the DNA will be something like this, double-stranded, but then it will be secular. It's a secular DNA molecule. So it's double-stranded, and then it's secular, like it's continuous like that. So how do these strands replicate? to begin with, it will start from in the sequence, the base pairs. You have a region which will be reaching A and T bases, adenine and thymine. So at this point, you have a molecule, you have a protein, DNA A. So you have this DNA A, it will come in and then it will bind at that region. And when it binds, it will break those hydrogen bonds between the A's and the T's. And this, that region, the A and T region, is what is known as the origin of replication. So you remember it's circular and there's this. So imagine it has been like that. But you have to remember that this DNA molecule is a huge molecule. And in order for it to fit in the nucleus, there'll be some twisting. It will twist around each other, something in this form, in order to fit in that. So when it comes to replication, those two strands have to be separated, and then you have a new strand forming on that, on one forming, and another strand forming on the other end. And so this separation will begin at the origin of replication, which is just a region which is rich in A and T base pairs. And DNA A protein, it's got some complex structure. Due to this structure it has will allow it to bind at that region, breaking the hydrogen bonds between the A's and the T's. And when it does that, you have one coming, then you have another DNA A molecule coming. Altogether, I think they bind to at the same origin of application. You have like about 8 or so of them bind, and that binding will cause the base pairs ahead of the region they've bound to. So they bind here. So they bind here. Then you have somewhere slightly ahead, the DNA molecule there will unwind. And when it unwinds, because it's already tangled up, you have it forming more tangles and so on. And because of that, you have the enzyme topoisomerase come in. What topoisomerase does is that, so what topoisomerase does, say, let me use this as an example. So you have the DNA molecule tangled up, and then what the topoisomerase will do is that it will break and then straighten it and then put it back together. So if I was to use this, here where it's tangled up, then imagine it tangles up some more due to the unwinding going on. So that doesn't, the tangling up sort of makes the molecule unstable. So the topoisomerase will come in, it will find where, it will come in where the intertwining is, and then break. And then when it sort of straightens up, seal up again. And so in the initiation, you have the DNA coming, the DNA A protein coming, bind to that origin of replication, which will then lead to further entanglement in the DNA molecule. There'll be more intertwining and so on. And because of that, topoisomerase comes in and then relieves those intertwining knots that are formed.
Hello? Kindly repeat the point. The meeting had cut.
So I was saying you have a circular DNA molecule, and this circular DNA molecule is a large molecule. So in order for it to fit in the nucleus, there's what is known as it's supercoils. So if you had an elastic band, say, and then you, those elastic bands, and then you try and fit it, say, in bottle top, you'd have to like twist it. I don't know if I'm trying to illustrate. You twist it so that it can fit there. That's what happens to the DNA. So because of that twisting, which is known as supercoiling, it poses a problem when replication starts, that in such a way that when you start to unwind one strand, you have, because of that unwinding, there'll be more coils that will be forming. And that coiling doesn't favor replication. So in order for replication to continue, you have topoisomerase coming, and topoisomerase, what it does is that it will cut where the coiling is, and then straighten, and then reseal the DNA molecule again. So the whole process starts with DNA binding to the origin of replication, which is a AT-rich region. And when it bound, topoisomerase comes in, and the end result is that you have a formation of an initiation complex, which will contain the DNA molecules, which are bound by DNA. And then ahead of that binding, you have topoisomerase, which is unwinding, preventing the supercoiling from happening. That's on the initiation. After the initiation complex is formed, you have DNA denaturation.
Excuse me.
Go ahead. Hello? So as I was saying, so after the initiation complex has been formed, you have this initiation complex here. Another enzyme will come in, helicase. This enzyme now, this enzyme comes in now to break the hydrogen bonds between the base pairs in the DNA molecule. And it does this using energy from ATP. And because it uses energy, if you remember from your structure of the nucleotides, you have a phosphate group attached to the 5' end. In order to achieve this, in order to acquire the ATP, it can only work from the 5' to the 3' direction. So meaning the unwinding will take place from the 5' direction going to the 3' direction. And then the helicase will go on like that, and zipping it will go on from the 5' end and zipping. That's from after you have the initiation complex, slightly ahead, the DNA unwinds, then helicase comes in, and then now starts breaking the hydrogen bonds there, separating the strands.
I read somewhere that the topoisomerase, it has two arms. There is one arm which does the cutting, and the other arm which like binds those cutting together after it it does the. Which two arms are those?
I don't think it's really important for you to, at this stage, to know the structure of the topoisomerase. Like when they say actually topoisomerase, there's more than one topoisomerase. In this case, this would be topoisomerase 1. Then you have topoisomerase 2, 3, and 4. So those differ in structure and in the way they sort of do their action, but the main thing they do is that they'll cut and then straighten, and then reseal the DNA again. I don't know.
Yes, maybe if you want more details, you can go search in Solomon Berg.
Mhm.
They have put more details in terms of the topoisomerase.
Okay. Yeah. Okay. So I was saying the helicase now has come in. It's broken the hydrogen bonds, and as it's breaking, it's hydrolyzing ATP from the DNA molecules. Now, these bonds that have been broken, they can again reform these hydrogen bonds. And so to prevent that, there's what are known as single strand binding proteins. These will come in and then they will bind to the single strands they'll bind to prevent them from rejoining and forming the double helix again, reforming the structure the way it was again. So once the single strand binding proteins are bound to the separated strands, the hydrogen bonding won't occur and there will be no reforming. Which brings us to the next step, which is priming. So you've separated the strands. Yeah, you've started the replication from the origin of replication. You've separated the strands. And then now, before the synthesis can start, what will happen is that RNA polymerase will come in and it will make a short RNA molecule. But why does it have to do that? The reason behind that is that the DNA polymerase cannot just come and start making a new strand just like that. The only thing the DNA polymerase can do is it can add nucleotides to an already existing strand. So it will come in and then it will add to the 3' end of the strand, and then it will continue adding on like that. But first, it needs that there's already a strand there. So RNA polymerase will come in, it will read this side, and then it will make a short RNA molecule. This short RNA molecule is known as a RNA primer. And then once this is formed, DNA polymerase will come in and it will continue the synthesis. It will do the same on the other strand, it will form, and then just like that. The synthesis of the two strands differs, and we'll explain as we go on to the elongation. So in the elongation, you've already formed that RNA polymer, that RNA primer, and then the DNA polymerase has now come in. So you have one strand, if you recall, due to the complementary base pairing, you have one strand which will be running 3' to 5', and then you have 5' to 3'. And then when these are separated now, the RNA polymerase comes in, and the strand it will make on this strand which runs 3' to 5', the newly synthesized strand will run 5' to 3'. So there you have the 3' end, which DNA polymerase like. So DNA polymerase will come now, and it will now start adding to that 3' end, moving on, synthesizing this side. On the other strand, the original strand runs from 5' to 3'. Then RNA polymerase will come in, and what it will make will be a strand which will be 3' to 5'. Polymerase will make a strand which will be 3' to 5'. But however, like I mentioned earlier, DNA polymerase can only add to the 3' end. It can't add to the 5' end. So this poses a problem for the synthesis of the other strand. And what will happen is that the DNA polymerase won't say since this is 3' to 5', I won't synthesize it. No, it will be synthesized, but the way it will be synthesized is that so you have this strand here, and then it will sort of bend backwards. So you have those two strands. One is running, so you have one running 5' to 3', and then complementary base pairing, you have this one running 5', anti-parallel with their model. So you have this original DNA, and then you have RNA polymerase coming on this strand. What it will make will be a strand that's 5' to 3', which is the RNA primer. Then DNA polymerase, which is able to add to the 3' end or continue with the synthesis and so on. On this other strand, what the RNA polymerase will make will be something which will be 3' to 5'. However, DNA polymerase can't add to the 5' end. So what it would do is that it would twist this molecule in such a way that you have it after it's twisted. You have that 5', 5', - 3'. And then what will happen is that the RNA polymerase will sort of twist it, bend it in such a way that you have the 3' end in the direction where it's synthesizing. You have it something like that, and then the DNA polymerase will come in and then synthesize. So you have this strand here, which runs 5' to 3'. The blue ones are the original strands. Then you have another one which runs 3', 5'. And then what you have is that during the synthesis, so here, if you can see my pointer, the RNA primer that was synthesized 5' to 3', then the DNA polymerase now continues with the synthesis going in that way. On this other one, since you have it running 3' to 5', the RNA polymer that is synthesized is 3' to 5'. But since the DNA polymerase can't add to the 5' end, what it would do is that it would twist it and then continue synthesizing on the 3' end. So in that twisting, when it's twisted, the original molecule is exposed again, and then the RNA primer will come in and then synthesize, and the RNA polymerase will come in and synthesize another RNA primer, and it will continue synthesizing just like that. So this brings about two different ways in which the strands are synthesized. So you have one which will be known as the leading strand, which is the one which runs 5' to 3'. This one will be synthesized in a continuous manner, whereas the other one, which runs 3' to 5', this one will be synthesized in a discontinuous manner because of the complex mechanism where it has to be bended, and then a primer has to be made and so on. And because of those primers being made, you have a situation whereby you have a lot of those primers. Those primers will be known as Okazaki fragments. And another thing that should be mentioned, the same DNA polymerases that are synthesizing these strands, they have an additional function apart from the synthesis, which is proofreading. So for this leading strand, like for the whole process, in prokaryotes such as the E. coli, the DNA polymerase will be DNA polymerase 3. It will be synthesizing. As it goes on, in the event that it adds a wrong base pair, it will change it and continue. Before continuing, it will change that base pair with the correct one. And then you also have, so you have DNA polymerase 3, then you also have DNA polymerase 1 and 2. So one, what it will do is on this ligand strand, you have these RNA primers that have been made. And then due to the twisting and all, you might have some spaces in between the fragments. So the DNA polymerase will come in and it will fill those fragments. Polymerase 2 is one which fills the fragments. Polymerase 1 will come in and it will change this RNA primer into a DNA molecule. And I've talked about the proofreading. I've talked about the elongation proofreading. Okay, termination. So as I said earlier, you have the secure DNA molecule, which has been synthesized, been unwound there. Let's say this is the molecule. It's been split as it goes, been split, been split, been split, been split. Eventually, it will reach a point where it meets, and then when it meets like that, you have two strands form. So it will cut there, and the two strands will fuse in such a manner. When they meet, so in this slide, the term used is forks, growing forks. So what is meant is simply this. So you have this structure here. You see the way it looks, and then you have a new strand forming there and another strand forming there. So this structure is known as a replication fork. So you have this side and this side. So as it goes on, remember that these two originals are still connected to each other. So when they reach the far end, and then they are cut there, and then they'll fuse, and then you have two.
Can you repeat, sir, there?
Yeah, on the termination.
Yes, sir.
Okay. So you have this secure DNA molecule. Say the replication started from here. So it will go on in one direction like that, continues until it finally reaches that point again. And when it reaches that point, and then they're cut, and then now you have two new DNA. So as it's cutting on this blue strand, a new strand is being formed. On the red one, a new strand is being formed. So it will continue just like that, continue until it reaches the end, the meeting point again. And then once it reaches that point, the replication is done. So once it reaches that point, you have two new strands, two new DNA molecules, I mean. Of course, there'll be, because of the secure nature, there'll be all that intertwining, but you have two DNA molecules.
Thank you, sir. It makes sense now.
Okay. So now you have the two strands. What's left now is to separate them. Like I said, the DNA molecule being used, there's that supercoiling and so on. And so when there's two strands are formed, they'll be intertwined. And then now you get topoisomerases come in again. In this instance, you have topoisomerase 2, which is also known as DNA gyrase, as topoisomerase 4. So these will work together. They'll say they come there where there's this intertwining, they'll cut one and then reconnect it on after untangling it, and they'll continue doing that. They'll continue untangling until you now finally have two separate DNA molecules. Okay, so describe the various stages of prokaryotic DNA replication. I believe we've done that. list the enzymes and non-enzyme proteins involved in prokaryotic DNA synthesis and explain the role of that. As we were talking about the stages, I also talked about the various enzymes, and I've put up this summary table. So you have DNA involved in the initiation process, binds DNA to form the initiation complex. We talked about it. We've talked about the topoisomerase and how it removes the coils and supercoils from the DNA. We talked about the DNA polymerase 1, which removes the RNA primers from the DNA strand. So it will remove the RNA primers and replace them with DNA primers. Then you have DNA polymerase 2, which I said after the whole process, it will go on the lagging strand because of the complex mechanism the lagging strand is synthesized in. You have instances where maybe there are some gaps in between the Okazaki fragments, and then the DNA polymerase 2 will go in and fill in. And then you have the DNA polymerase 3, which is responsible for the main DNA polymerization. But what you should know is that all three of them have some proofreading activity. So DNA polymerase 1, before continuing on with the polymerization, if it makes a mistake, it will try and correct that mistake. And all this, as they are cleaning up, if they find mismatched base pairs and so on, they will change those. And then I talked about helicase, which is involved in the DNA denaturation. It simply breaks the hydrogen bonds using ATP for its energy. And then you have primase, which I've been referring to as RNA polymerase, which simply just makes the short primer before the DNA polymerase can come in and continue with the synthesis. Then you have ligase, which joins the DNA nucleotides through covalence. And then I also mentioned the single strand binding protein. That was from the denaturation step. After helicase separates the strands, you have there's a chance that these bonds can form again, the hydrogen bonds, and to prevent that, the single strand binding proteins. So I've explained on the enzymes and non-enzyme proteins that are involved in prokaryotic DNA synthesis. As well as explaining the various stages, I also explained on the leading and the lagging strand and how the two strands are synthesized differently. You have the leading strand, which will be synthesized continuously because it runs in the 5 to 3 prime, the same direction in which the DNA polymerase is able to work. And then you have the lagging strand, which through complex mechanisms will be synthesized by the same DNA polymerase, but instead this synthesis will be discontinuous. For the leading strand, you have it, you only have it, the RNA polymerase will only need to add one RNA primer. Only need to add one RNA primer, and then the DNA polymerase will continue. But for the lagging strand, again, due to the complex mechanisms I explained in the previous slides, you have multiple RNA primers. Then you have sections where after the DNA polymerase has synthesized, so you have that complex where you have RNA primer connected to a DNA nucleotide, and then you have another RNA primer connected to a DNA nucleotide. Those RNA DNA nucleotide molecules are what are known as Okazaki fragments. So in the lagging strand, you have formation of Okazaki fragments. The last question, which says explain how accuracy is achieved during DNA replication. I explained that when I was talking about the elongation and the proofreading activities of the DNA polymerase. To bring us to the end of our discussion on DNA replication. I hope it's been helpful. Thank you very much. Hope to see you in the next session.